All right, I'm just going to start by recapping what we already did in Lesson 10. So this is all from Thursday's class, so I'm not going to spend a lot of time on each slide. We started off talking about glucose, what happens to glucose, and I want you to really recognize that this is the glucose that we just made in photosynthesis. Now we're using it, and we're going to basically take it apart. So we started by writing out the reaction. It wasn't new because we already wrote the reaction one of the very first lessons. Maybe what I want to emphasize is the part where there were 36 ATP. The fact that there are 36 is actually fairly important because I'm going to show you where all 36 of them come from so that you can see that it adds up to 36. We also talked about eventually how I'm getting glucose to all of my cells. And so recognize every cell of your body, maybe with a few exceptions, but every cell of your body needs to do cell respiration, otherwise it dies. So even when you're just sitting here, you know, tired, it's morning, you're not really moving around, you're still doing cell respiration. You don't have to be exercising for you to require glucose to function. You have what's called a basal metabolic rate. It's something that fitness trainers measure all the time to determine how many calories you should eat per day, etc. That's how much energy you need if you just laid there awake, not moving, doing nothing for the whole day. And most of you need over 1,000 calories a day just to do that. So cellular respiration happens whether or not you do nothing during the day, whether or not you run 3 marathons. Then we mentioned where glucose goes. This, of course, being the idea that we're going to focus on now, the production of ATP and heat. The other things, remember, were ways that you could store glucose or, of course, get rid of it. And remember that I mentioned it would be incredibly rare for you to have glucose in your urine. If you did, it probably means that you have or that you're diabetic or pre-diabetic perhaps, and that's a test that they actually do for that. And then we ended up here, and I showed you a video of a guy singing a super awesome song about cell respiration that I would encourage you to watch. Watch that video as part of your studying for the cell respiration part, because it's quite catchy, and it might help you remember some of the pieces that are happening. This is the overview. Glucose oxidation is what is happening during cell respiration. So in photosynthesis, I took CO2, I reduced it, I made glucose. Now I'm just going to undo that. I'm going to oxidize glucose and get my CO2 back. So we are going to remove hydrogen and electrons from glucose, which are the things that we added to CO2 to make glucose. And we're going to get CO2 back. Now, you'll see that there are some words here, and I'm just going to explain what's happening. This little chart illustrates different pathways that cell respiration can take. They all start with glycolysis. So glycolysis, and if you look at the word glycolysis, I'm cutting my glucose. That's literally what the word means. I'm going to take it, I'm going to cut it in half. So there's glycolysis, and that's the first step no matter what. All organisms of any type can do that step. You could be a bacteria, you could be a piece of corn, you could be a cow, you could be a human, you could be I don't know, a spider, anything. All living organisms do this part because this part occurs in the cytoplasm. That means I'm not inside the mitochondria yet. So even cells that don't have mitochondrion can do this part. Now, depending on whether oxygen is absent or oxygen is present, we have a couple of pathways. If there is no oxygen available, we end up doing something called fermentation. typically. Fermentation is the way that you get acids and alcohols produced. For example, in your muscles you have lactic acid. If a yeast were to perform this, I might get alcohol, which is how alcohol is made. I take an organism and I ferment it. If oxygen is present, then I move into this box, and this box is supposed to represent a mitochondrion. So the first step happens outside of the mitochondria. Then I move into the mitochondria if I have one. If I don't have a mitochondrion, I can't do those two parts. So we have glycolysis, then the Krebs cycle, then the electron transport chain. Those are the three basic parts of cell respiration. And then this would be like step 2A. If I can't do the Krebs cycle because I don't have a mitochondrion or I don't have oxygen, then I would move on and do fermentation instead. So this tells you the overview. We're going to go through and talk about the details of each of the parts, and we're going to start with glycolysis. Does anybody want to ask any questions here? So the key idea really from this slide is that you recognize that every organism can do that first step, and that only if you have a mitochondrion can you move on to step 2 and step 3. So, glucose oxidation number 1, and I keep calling it glucose oxidation because I want you to remember that's what we're doing. We are taking glucose and we are oxidizing it. It's called glycolysis. I have a picture of it on the side, and I'm going to explain what you need to know from the picture. It's important to me that you know that it starts with glucose. Now, what happens in glycolysis, which occurs in the cytoplasm and does not use oxygen, like I mentioned earlier, is that I take one glucose, something that has 6 carbons, and I end up splitting it into 2 pyruvic acids at the end. Each of those have 3 carbons. I'm showing you the number of carbons so that you can see I'm cutting it in half. I'm breaking it into 2 pieces. Now sometimes pyruvic acid is called, let's use a better color, is called pyruvate. That's the name of the substance before it's dissolved in water. You might see both terms used. I try to use pyruvic acid as much as I can. So you need to know what it starts with and what it ends up with. The names of these substances right here, I won't ask you, although I will show you this. That's the thing that you made in the Calvin-Benson cycle. So we're just taking glucose and undoing the Calvin-Benson cycle. Okay, but you don't need to know the names of those intermediary substances. You need to know the name of what I start with, what I finish with. You also need to know in terms of energy molecules what's produced. So I have 2 pyruvic acid, we saw. I also get 2 NADHs and 4 ATPs. So I'll show you 1, 2 NADHs. So NAD is like NADP that we used in photosynthesis, same kind of molecule. It just doesn't have the phosphate group on it, so we don't use the P. When it doesn't have the H, it's oxidized. When it has the H, it's reduced. So this got reduced. That's how I made NADH. If it got reduced, glucose had to be the one that got oxidized. So remember, if something gets oxidized, something else can be reduced, but they have to happen together. Now I also get, technically, 4 ATP. 2 there, 2 on the other side. But if you read the next point, or you remember in the song, the guy says, 4 ATP are made, oh, it's really 2. Here's why. Way back at the beginning, up here, I had to use 2 ATP. So before this process could even start, I had to have 2 ATP already made and use them to get the process going. Basically, I'm using it to phosphorylate glucose. That means I'm adding phosphate to glucose. It makes it more active, and it makes it so that glucose cannot diffuse out of the cell. It makes it so that it has to stay and do this process. So, if I make 4 and I use 2, the net result is 2 ATP. And that's a fairly key idea. If I take this whole process and I ask myself, what's the difference between what I had at the beginning and what I have at the end? At the end, I have 2 more ATP than I started with. Now, I actually made 4, but because I used 2 at the beginning, the net result is that there are 2. Now, is there any part of maybe the diagram or something that's written that anybody wants to ask about? So much like with the parts of photosynthesis, either the first or the second part, you'll notice it's not the little details that we're looking for. We're looking for the big main idea. As long as you can tell me what I start with, what I finish with, and any energy molecules I make along the way, that is the key idea that you need to know about glycolysis. Now, please don't forget, it occurs in the cytoplasm, not the mitochondria, which means any organism who is alive and has cytoplasm, which is any living cell, should be able to do this part. Now, what that means is that if you don't have a mitochondria, you're fairly limited as to how much ATP you can make. We're going to make 2 ATP here, but I said that there were 36 in the overall cell respiration reaction. So obviously, if you're only doing this first step, you're not making a lot of ATP. You're only making a little tiny amount. Okay, so this is glycolysis. What I'll show you next is what are some choices for that pyruvic acid at the end? Where could it possibly go after it does glycolysis? And like we saw, it's either going to go into the mitochondrion or off to some other process. So, what happens to pyruvic acid? And I'll just remind you, it can be called pyruvate. We're talking about the same thing. One choice is anaerobic respiration. So we're going to talk about that word. When you have a or an at the beginning of a word in science, it means not or no. And aerobic means oxygen. So one of the choices is that pyruvate, that pyruvic acid, will be in an environment where there is no oxygen. Now, fermentation goes under this umbrella. Technically, fermentation and anaerobic respiration are not identical. Anaerobic respiration is usually what we call it when the organism doesn't usually have oxygen. They live specifically in an environment without it. Whereas fermentation is what happens when an organism that normally has oxygen is without it. It's a fairly slight difference, and it's not something that I would typically ask about. But just so you know, they're almost interchangeable. Maybe we'll say fermentation is a type of anaerobic respiration, but it's the one that we're going to use. So for us, we're talking about glucose, which we know we just turned into pyruvate, is going to become lactic acid or alcohol. Those are the two cases that we would care about. Now, the lactic acid part occurs in muscle cells. And it only occurs during really strenuous exercise. So if I ever ask you on a test, when does anaerobic respiration happen in you, you could say in your muscles during strenuous exercise. What's happening is that the cell uses oxygen faster than the circulatory system can provide it. That means that your cells would prefer to use oxygen, but there's not enough of it. A similar process occurs in yeast cells to produce alcohol. So if you've ever been around when someone is making beer or making wine, you might know this, but you have to put it in a container without oxygen. And that's how you get your fruit or your yeast cells or whatever kind of grains you're using to ferment and produce alcohol. Now the next point is pretty important. Not all cells in your body can do this. So not all of your cells can do anaerobic respiration. Some cells in your body die. Brain cells would be an example. Your brain cells do not do anaerobic respiration. If they don't have oxygen, they give up. They die. Now we'll focus on the thing that's happening in you, in people, because later on we're going to talk about human physiology, so we want it to be related to what we're going to talk about later on. Lactic acid, which is what comes from your muscles, decreases pH. There's a point. Acids make the pH lower. I frequently get people telling me that acids make the pH higher because you think if I add an acid, I should be adding something. But remember, lower numbers mean stronger acids. Your muscles cannot contract if the pH gets too low. And in our next chapter about muscles, you'll see why. How do I overcome muscle fatigue? When the oxygen debt, that means the oxygen that I wanted that my circulatory system couldn't provide, when I finally breathe hard enough, when my heart beats fast enough to bring the amount of oxygen necessary to all of my muscle cells, so there's increased breathing, increased heart rate. Those are two important things because those are the physiological signs. So I can't see that you're doing anaerobic respiration, but I could see that you are breathing hard. You could feel or hear that someone's heart was beating really fast. So those are measurable things that we could use, say, as part of a lab experiment. And we will, in fact, use those as part of a lab experiment later on. But when those two things increase so that the supply of oxygen increases, then my cells don't have to do this anymore. So for our cells, humans, we would prefer not to do anaerobic respiration. It's a second choice when we have to, when there's not enough oxygen available. Now what happens? Lactic acid is converted into glycogen. Now we've heard of glycogen before. It's the storage form of energy in your muscles. So lactic acid isn't totally useless. It's doing something. This conversion gives me 2 ATP. So I made 2 in glycolysis, and then I made 2 more here. And if anaerobic respiration is all I was doing, I would have a total of 4 ATPs. So I'll just show you up at the top here what's happened so far. This bit here is glycolysis. It's a repeat. It's showing me that I use 2 ATP to phosphorylate glucose. I oxidize glucose. I end up with pyruvate acid and 4 ATPs. So that just sums up glycolysis. Then we're showing that once I have the acid, if I'm a yeast, I would be making alcohol. Homolactic fermentation, Homo sapiens, that's us, we're humans. So that's what happens to us. We produce lactic acid, and I get 2 ATP from that. So you'll notice there's no intermediate compounds. I'm not expecting you to know the detailed steps of how each thing is produced, just what happens in the long run. So if I do anaerobic respiration, 4 ATP total is all I can possibly have. 2 from glycolysis and the 2 that I'll get doing this process where I take lactic acid and convert it. Now, does anyone want to ask any questions, perhaps about lactic acid or muscles? One of the things maybe that I will mention, and we'll read an article about it when we talk about muscles, a lot of people think that when your muscles hurt the day after a really hard workout, it's because of lactic acid. Your body clears lactic acid, hey, as soon as you're done breathing hard. Lactic acid is only there if your body is doing anaerobic cellular respiration. So a lot of people have this misconception that your muscles hurt because you're still doing anaerobic cellular respiration. That's not why. Your muscles hurt because you tore them when you exercised, and that's where the pain is coming from. If you still were doing anaerobic respiration 24 hours after you worked out, you would be dead, because that would mean you did not have oxygen available for like 24 entire hours. There's no way your brain tissue could have survived that long. All right, so that is the first thing that can happen to pyruvic acid. It does anaerobic respiration, meaning no oxygen present. This part all happens in the cytoplasm, all of it. So organisms that don't have mitochondrion can only do this part. They can't do the next part that happens in the mitochondrion. So what's my other choice? My other choice is aerobic respiration. Now on the left, this is the same diagram that was just on the screen, but I uncovered the last little part of it so that you could see the other option for pyruvic acid. It's moving across a membrane here from the cytoplasm into the mitochondrion. So if a cell has oxygen available and it has mitochondria, then it can do aerobic cellular respiration. If either one of those two conditions are not there, this won't happen. Now the basic idea, if you look right here, is that we add oxygen, because remember, we are oxidizing things. And one of the reasons it's called oxidizing is because historically, oxygen was a substance that was really good at oxidizing things. And somehow over here, I am going to take 34 ATP, or 34 ADP, sorry, and make 34 ATP. I'm also going to produce CO2 and water, which makes sense. That was what I used to make glucose in the first place. Now, aerobic respiration technically has 3 steps. Glycolysis, which happens first, and we already talked about, is the first step. Even though you don't need oxygen for that part, if glycolysis doesn't happen, you can't do the other 2 parts. The Krebs cycle. Now the Krebs cycle could also be called the citric acid cycle. I would imagine that Krebs is the name you would see more frequently, but just in case, that's another possible name for it. And then the electron transport chain. And the last two parts are the ones that are happening inside the mitochondria. So far, we know what happens to glucose. It gets split in half to make pyruvic acid. And we know that pyruvic acid has two choices. Either there's no oxygen or no mitochondrion, and we stay over here, and we do anaerobic cellular respiration, or there is oxygen, and there is a mitochondrion, and I can move on and do aerobic cellular respiration. Now, just a little bit of vocab here. When something uses or doesn't use oxygen, we're talking about the type of respiration that it's doing. If I want to talk just about the environmental conditions, you'll see a couple of words that are different. Oxic and anoxic. Oxic means there is oxygen, anoxic means there is not. It's a small point, but that's when we're talking about just what is available, not what the organism is actually doing. Now, does anyone want to ask any questions up to here? All right, so we will talk about the other two parts, the Krebs cycle and the electron transport chain, in our class tomorrow.